And I'll put my computer over where it'll pick you up. Can you hear Tim now?
Can you hear me? Okay. Sorry about that. Okay, so yeah, this function can promote several disease states. So a couple examples of Rett disease. I still can't see anything. There we go. So with cases of loss of function mutations, Rett can develop into Hirschsprung's disease. So without that Rett activation, we get kind of interrupted innervation of the gut. However, for the purposes of this talk, obviously we're going to be focusing on Rett's role as a proto-oncogene. So in cases of gain of function mutations, Rett is responsible for the tumor syndrome MEN2, which is characterized by thyroid cancer and pheochromocytomas. On the other hand, we're also interested in the expression of just the wild type Rett receptor, which is expression is observed in several different cancers. However, those are cases where Rett is not the driver, but may be contributing to these tumors, including breast or pancreatic cancers, as well as pheochromocytomas again. So before I get into my specific projects, I'm going to delve just a little bit deeper into the process of Rett activation formation. So Rett is made in the endoplasmic reticulum at about 155 kilodaltons, or the immature band on a Western blot. It's post-translationally modified to 175 kilodaltons, or the mature protein, which then progresses to the cell membrane and can be activated in the presence of its ligand and its co-receptor. Its co-receptor GFR alpha is localized within lipid rafts. So that kind of recruits Rett to these lipid rafts, which are kind of signaling platforms for many signaling pathways, including PI3K, like KT. But Rett can also signal through RAS, MAPK pathways. And these pathways are responsible for many cellular processes, including proliferation, survival, migration, and invasion. So some proteins of note are ERK, AKT, and S6, and they'll come up a little bit later. Once Rett is active, Rett is internalized into the cell through clathrin-mediated endocytosis, or CME. And this is a process where an active receptor recruits the adaptor protein AP2, which can then further recruit clathrin, or this lattice protein. And as you can see in this image, they create this lattice structure that brings some curvature to the membrane and pull these proteins into the cell. So this one's a little bit deeper, and then that's the clathrin-coated pit, and then the clathrin-coated vesicle will start to pinch off of the membrane, and that diamond too will cleave the membrane, and it'll float away off the membrane, and then this lattice structure will fall apart, and these proteins can enter the endolysosomal system, beginning in early endosomes. And then they can either be recycled back to the cell membrane or progress down to late endosomes and lysosomes for degradation. So as an outline of my projects and this presentation, I'm first going to look at the wild type receptor in a model of TMEM mutant pheochromocytomas. So we're going to look at TMEM's function itself, as well as the effect of Rett mislocalization on the activity of these cells. And then following that, we'll go into characterizing the MEN2 Rett mutants, looking at their subcellular trafficking, as well as some current theories and future directions I'm going to be looking at. So the TMEM project is titled Loss of Tumor Suppressor TMEM127 Drives Rett-Mediated Transformation Through Disrupted Membrane Dynamics. So for a little bit of background on PCCs and TMEM, pheochromocytomas, or PCCs, are rare neuroendocrine tumors of the adrenal medulla, and they're primarily driven by somatic or germline mutations in one of the several susceptibility genes. And two of these genes are Rett and TMEM, and these are both included within the kinase signaling cluster of these genes. TMEM127, for example, is a tumor suppressor gene, which has a suggested role in protein trafficking. But when we began this project, the data wasn't very solid, so we weren't really sure. So we want to investigate what exactly TMEM127 is doing. But something interesting we found was in cases of the TMEM mutant pheochromocytomas. So this IHC here includes pheochromocytomas with mutations in TMEM or RET itself, as well as SDHA or unknown mutations. And as you can see here, that these are stained for RET. And as you can see in the TMEM mutation cases, the RET expression is quite high, if not comparable to the RET mutation itself. So this high amount of RET expression in TMEM mutant pheochromocytomas, which was very interesting to us. And we wanted to investigate kind of what effects this high expression of RET could be having on these tumors. So getting into our cell model, here we used SHSY5Y cells, which are neuroblastoma cell line, because they endogenously express RET as well as TMEM. And then we have a TMEM knockout variant of them, so they don't have any TMEM. And we noticed a similar increase in RET expression. And then using a biotinylation assay to biotinylate all the cell surface proteins and then immunoprecipitate them, we show that this protein was biotinylated, meaning it's on the cell surface, and then there was about a 5-fold increase in RET on the cell surface in these cells. So RET was accumulating on the cell surface when TMEM was lost. However, this wasn't exclusive to RET. So we looked at EGFR, another receptor tyrosine kinase, as well as some cell adhesion molecules, as well as a carrier protein. And we see the similar pattern. So TMEM is lost, and we see an increase in expression of these proteins. So that kind of signified to us that it wasn't isolated to RET, but we're going to use RET as an example. But potentially there's some global defect going on when TMEM is knocked out. So looking specifically at just the effect of TMEM receptors, we looked at the formation of clathrin-coated pits, so that part of that clathrin-mediated endocytosis. The formation of these pits is a complex process. So to begin, you get kind of little clusters of AP2 and clathrin. And if they build up enough clathrin, they're able to kind of nucleate at a site of an active protein. And if you accumulate enough clathrin, you can then initiate a CCP formation, starts to curve the membrane. But if you don't accumulate enough clathrin, then these structures will just fall apart. And we call that kind of a failed nucleation step. So to look at this process, we used a form of microscopy called TIRF, or total internal reflection fluorescence microscopy. So as opposed to a traditional confocal, where the laser goes straight through the cell and you see all levels within the cell, this TIRF shoots the laser on an incidence angle. So you only excite about 90 nanometers into the cell, which is roughly everything at the cell membrane. So these videos are taken with TIRF. And as you see, the proteins come into focus, they're coming up to the membrane, and then they go away from the membrane and disappear. So ultimately what this showed are mock knockouts versus TMEM knockout cells. We see an increased proportion of these failed nucleation steps when TMEM is lost. So these little structures are forming and then falling apart and not actually initiating CCP formation. And therefore, we see a decrease in those CCP initiation events. So we see kind of less efficient initiation when TMEM is lost. Additionally, when we look using structured illumination microscopy, we can look at the structure of these CCPs. And we found that in the TMEM knockout cells, we see a decrease in size of these pits, as well as a decrease in the number per cell. So clearly the formation of kind of large protein complexes was less efficient in these TMEM knockout cells. We then went to look at the diffusion of another receptor, just kind of get a gauge of how this membrane was behaving. So we looked at the diffusion coefficients of EGFR, and we found that the EGFR showed increased diffusion. So essentially the membrane was more dynamic and these proteins were moving out around more. And then we went to look at, in the vein of kind of the membrane having more energy and these proteins zipping around, we looked at lipid raft or membrane microdomain sizes. And kind of consistent with this increased energy, we see a decrease in the average size of these lipid rafts, suggesting that they're less able to coalesce together. So ultimately, we came to the conclusion that TMEM seems to have some function as a membrane stabilizer. So you get rid of it, everything's zipping around, small protein complexes can't form. And we then kind of identify this as a useful tool to look at RET surface accumulation. So we lose TMEM, we see an accumulation of proteins, including RET. And essentially this simulates an overexpression. So if these proteins are accumulating, they are overexpressed in a sense. And we know that in the context of other receptors that accumulate on the cell surface, even in the wild type form, we do see some constitutive activity, just happenstance bumping into each other. So this model allowed us to explore what RET on the surface does to these cells. A tool to isolate RET to the cell surface. So looking at RET specifically now, so back to this RET and clathrin-coated pit area. This again is turf microscopy, so everything is at the cell membrane. In the mock knockout cells, we add ligand and we see an increase in RET going to clathrin structures. So these white arrows indicate areas of overlap of the two channels. So we add ligand, we see an increase of RET going to clathrin pits. However, irrespective of ligand treatment in the TMEM knockout cells, we see significantly less RET within these clathrin structures. Interestingly, we do see interaction with the adapter protein. So if anything, significantly higher amounts of that interaction in the TMEM cells. So they're interacting with AP2 and attempting to recruit to clathrin, but that step is non-functional. So kind of consistent with the increased membrane diffusion, RET isn't being kind of properly loaded into clathrin-coated pits. So going kind of the next step down from localization with clathrin, we looked at protein internalization. So we again labeled with biotin and then allowed for internalization. And then we stripped the surface, so any biotin left is signifies protein that had been internalized into the cell. And we see that in the TMEM knockout cells, we see less robust internalization over time, seemingly a background amount, so it's higher by 5 minutes, but not increased with longer treatments of ligand. So this is consistent with kind of less RET going to clathrin-coated pits, less RET is entering the cell through internalization, and then accumulating on the cell surface. And consistent with less RET coming into the cell, less RET is then able to enter the endolysosomal system and be degraded. So we see a decrease in RET degradation over time in the TMEM knockout cells, also meaning an increased half-life of these proteins. And then to go along with the idea that all this wild-type protein is stuck on the cell surface, we see some constitutive activity of these receptors. So we see constitutive phosphorylation of RET, as well as AKT, ERK, and S6. And here I have it graphed. So we see a significant increase in the amount of constitutive activity. And then with the idea that these cells are potentially insensitive to longer treatments of GDNF, we don't see a significant increase when we add ligand. And then we looked at whether this constitutive activity was actually RET mediated. So we tested bandetinib, a multi-kinase inhibitor, as well as 2 RET selective inhibitors, selpercatinib and pralsetinib. And in all the cases, we were able to turn off that constitutive activity, showing that these signals are RET mediated. And these signaling proteins are involved in these pathways, which are commonly associated with cell proliferation and other cell processes. So then we looked at cell proliferation, or relative cell viability as a marker for proliferation. And in the TMEM knockout cells, we see with or without ligand, we see a significantly increased amount of proliferation in these cells. However, when we introduce those RET inhibitors, well, selpercatinib is one of the RET selective inhibitors. We were able to not only knock back proliferation, but we also saw a 20% reduction in cell viability, suggesting that these cells with lots of RET on the cell membrane were potentially sensitized to RET inhibition. So going along with that, in collaboration with a lab in Texas and Dr. Patricia Dahia, we performed some treatment with sulfatinib in mouse models. So as you can see, when sulfatinib was introduced, we saw a significant decrease in the tumor volume and weight of these tumors. So in other words, potentially these sensitized cells are introduced to RET inhibition and they're prone to their viability is reduced and these tumors start to shrink. So ultimately what this suggested to us was that RET inhibition could be an effective treatment against TMEM127 mutant pheochromocytomas. So here I have kind of summarized all the findings into one figure, but ultimately this data indicated that kinase inhibitors could be useful against treating TMEM mutant tumors. So any of these tumors could be accumulating receptors on the membrane, and then you could use an inhibitor to try and counteract that activity. Additionally, we found that the localization had a very significant impact on the activity of the wild type RET protein. So going forward, we wanted to look at how the localization of RET mutants could be impacting their activity. So that leads me to my second project, which I titled Characterizing the Trafficking and Signaling of MEN2 RET Mutants. So for a little bit of background on multiple endocrine neoplasia type 2, again caused by these RET mutants. It is an autosomal dominant inherited tumor syndrome, and it's characterized by medullary thyroid carcinoma, as well as pheochromocytoma, so for both subtypes. However, there's some subtype-specific additional symptoms, including parathyroid hyperplasia for 2A, and 2B has mucosal neuromas of the mouth and tongue, as well as marfanoid body habitus, which is kind of extended growth of limbs. So MEN2A is less aggressive and has a later age of onset, but it's the most common of these inherited tumor syndromes. And for any immunology fans, the thyroid TME in these tumors is immunologically hot, so cytotoxic cells infiltrate fairly well. MEN2B, on the other hand, is more aggressive. It's far less common inheritedly, but and has far earlier onset. So it's less common inherited, but it's virtually the only form of sporadic that shows up. And again, for comparison, the thyroid TME is immunosuppressive and enriched in epithelial mesenchymal transition genes. Additionally, MEN2B has a more pronounced response to inhibition. So again, sulfatinib and pralsetinib are FDA-approved drugs for treatment of MEN2 in the thyroid. However, RET inhibitor resistance is a problem, as with many receptor inhibitors. So there is a need for some alternative approaches. And the kind of question we approached this with was, can we identify how and when to deliver drugs more effectively by understanding the behavior of these RET mutants? So for a little bit more information on the mutants themselves, MEN2A RET has mutations in the extracellular domain of RET. So C634 mutations are the most frequent. These occur in the extracellular domain, and they cause constitutive dimers of these proteins. So they're constitutively dimerized and active receptors. MEN2B, on the other hand, has a kinase domain mutation, which a tenfold increase in the activity of the kinase. So again, they can be constitutively active either as monomers or dimers. And then just as a reminder of how oncogenes work, one mutation or one copy mutated is enough to cause disease. But that also means that there's a wild type copy still remaining, except for in some rare cases where homozygous mutations have been seen, but those are largely in consanguineous families or families of parents that were closely related. So as Dr. Greer mentioned, Lois and I worked on a review paper this summer where we were kind of looking at all that we currently know of these MEN2 mutants. And we kind of boiled them down into intrinsic and extrinsic properties. So intrinsic are within the protein themselves. What do we know about the proteins? And then extrinsic is kind of everything around it, where the proteins are, where they're signaling. So as far as the intrinsic properties, these are very well studied. So the mutations are known, the mechanism of activation of all the phosphotyrosines within the protein, as well as their dimerization status. And ultimately, both receptors are constitutively active, but it's difficult to tell any difference beyond that, just based on those. So extrinsic properties are where our focus lies. There's some less established and quite old. A lot of the studies were in the early 2000s, and not much has been done since. So there's some data on the trafficking and signaling localization, but often these studies tackled one mutant or the other, and compared to the wild type. So this is where we aim to look, and we want to look to see if we can identify any differences or potential areas for targeting, any differences between 2A and 2B, or just differences of them to the wild type. And we first wanted to look at, investigate how the trafficking and localization of these receptors differ. So a quick mention of our cell models. The majority of these investigations were using the endogenous cells. So we have SHSY5Y cells. Again, these are a neuroblastoma cell line, as well as the MTC-TT cells, or just TT for short, and the MZ-CRC ones, which are just termed MZ cells. So the TTs and MZs are derived from medullary thyroid carcinoma. However, there's not really an available cell line with wild type RET derived from C cells. So we are using these neuroblastoma cell lines as a proxy for that, because they express wild type RET. Additionally, we have some ADVEC cells that come in at the end. These are SY5Y cells, which have a RET knockout, but then we added back wild type 2A or 2B. And these are advantageous because they are the same cell background, so it's a kind of a direct comparison of the RET forms. But again, the majority of this was done in the endogenous cells. So starting off, looking at kind of an overview of the trafficking. Again, this is a more complicated version of the biotinylation assay, but ultimately we label all the cell surface proteins, we allow for internalization, and then we can extend this by allowing an extra 15 minutes for recycling back to the membrane. And then this version does the same thing, but we strip the surface. So then the amount of protein recycled is the difference between DC and R15. So I have that the percent recycled is under here. And ultimately what this showed was by 5 minutes of internalization, there's no significant differences. However, by 30 minutes, both MEN2 mutants have significantly increased internalization into the cell. And then the extra 15 minutes after that for recycling, we see that both MEN2A and B are significantly increased over the wild type, and 2A even more so than 2B. So 2A recycles constitutively more than the 2B mutant. And then kind of moving back to that first 5 minutes of internalization, we looked again using TURF microscopy. We looked at RET colocalizing to clathrin. And similar to the TMM work, we add GDNF or ligand to the wild type cells, and we see an increase of RET going to clathrin. And then if you look at 2A and 2B, they're significantly increased even beyond that wild type treated with ligand. So these proteins are constitutively localizing to clathrin structures. Additionally, the puncta intensity on the cell membrane of these receptors is significantly increased. So there's quite a bit of protein on the cell surface with these 2A and 2B mutants. And then going to the 30-minute time point. So I've indicated here. So these proteins are going to the clathrin pits and then they internalize. So we are looking at EEA1, or the marker of early endosomes. And we see that both 2A and 2B are significantly increased presence within these early endosomes, suggesting that they're internalizing far more. And then as a quick aside, this is another cell model. These are RPE cells transfected with wild type 2A or 2B, just because kind of as a confirmation of this EEA1 internalization. And we do see the same pattern. So 2A and 2B are present within these endosomes. We also tested the late endosome marker, so the next step after that. And we see that 2A and 2B are constitutively co-localizing with early and late endosomes. This is the only slide where this model was used. So back to the endogenous cells, we then looked at, so we know that they're going early endosomes and late endosomes, and then we wanted to check whether they're recycling. So consistent with the recycling we saw up here, we do see a significant amount. Again, this is a preliminary study, but we see a significant amount of co-localization with recycling endosomes. And then going on from that, we wanted to look at the degradation. So we know that these proteins are in all of these endosomal compartments. And then the last step in that would be the compartments that degrade the proteins, or the lysosomes. So we looked at protein degradation over time. So again, these cells were treated with cyclohexamide to stop protein synthesis and just watch the protein degrade over time. And we see that the wild type cells degrade quite noticeably. However, the 2A and 2B have significantly reduced degradation, but also the difference between them is somewhat proportional to the recycling. So the 2A has the least amount of degradation and the most amount of recycling. So it's recycling and degrading the least amount, and 2B is somewhere in the middle. So then looking at the signaling activity, so these receptors are trafficking within the cell and back up to the membrane. We wanted to look at the signaling of these proteins, and this is in the ADVEC cells. Given that it's a more direct comparison of RET forms, we figured this was adequate for the signaling. So in the wild type cells, we add ligand and we see an increase in RET phosphorylation. And then 2A and 2B, as we would have predicted, we see quite a bit of RET phosphorylation. Again, this is preliminary. I need one more replicate to finish this, but we also see constitutive ERK signaling as well as S6 signaling. But one interesting thing, given that these receptors are constitutively active, we don't predict that they would really need the co-receptor. And given that that co-receptor is isolated within lipid rafts, they shouldn't have any connection. They shouldn't really need to go to lipid rafts for activity. And we don't see significant AKT phosphorylation, which is kind of a lipid raft isolated signaling pathway. So this suggests that these proteins are outside of lipid rafts and not signaling through AKT, which is interesting. And the phospho-ERK is consistent. It's often activated intracellularly. So that would be consistent with the trafficking, the high levels of RET phosphorylation. So here is a little summary. The size of these RETs is representative of the amount of RET in each place. So we see quite a bit of 2A and 2B are present in clathrin-coated pits and vesicles, and then within early endosomes. And then we see a slight difference, or a significant difference, in recycling. However, when you get down to the lysosomes, there's a significant decrease in the presence of 2A and 2B, so they're not degrading. So that's the kind of summary. And then there was a bit of data that didn't really fit the rest of the story. but I thought it would be interesting as to lead me into my future directions. So these are the SY5Ys, TTs, and MZs. And all of this work was done without ligand to kind of see what the constitutive activity of these proteins was doing. But we also tested to see the co-receptors that were present in these cells. And we found that, so I mentioned that GFR alpha 1 was the one I've kind of been concerned with the majority of the time, but this is kind of a family of GFR alpha members, and they have kind of distinct and overlapping expressions. So we found that SY5Ys, they have GFR alpha 1, but they also have GFR alpha 2 and 3. And then TT cells have GFR alpha 1 as well as 4. But interestingly, the MZs don't really have any of them. And I found this interesting because, so as I mentioned before, the wild type RET comes into contact with its ligand and will dimerize with the co-receptors, and then it's kind of brought into the cell and regulated. The 2A RET receptor, on the other hand, again, as I mentioned, these cells still have a copy of the wild type receptor. So we were curious as to whether this wild type RET could be regulating Men2A RET through kind of heterodimerization. So I mentioned that they're constitutively dimerized, but not every protein is immediately dimerized by the time it reaches the membrane. So it could be binding the wild type receptor in the presence of ligand, and that could be partially regulating Men2A RET's activity. Whereas Men2B, with no visible co-receptor, addition of the ligand presumably wouldn't be recruiting wild type RET, which then couldn't recruit 2A. So 2B would still be regulating itself on its own and kind of cycling through the cell. So that kind of leads me into my burning questions or next steps. So the first thing to kind of test this would be expanding the cyclohexamide assay. So I wouldn't predict that adding ligand to the 2B cells would affect them very much because there's no co-receptor. So they're probably doing the same thing they were before. However, the 2A, if it's partially regulated by the wild type, adding ligand would be binding the wild type and internalizing. So I would predict to see a decrease in the degradation, or a decreased half-life. Additionally, with the idea that there's no phospho-AKT, signifying that there's probably no presence within this lipid rafts, I'd also like to try some TERF, again, imaging at the cell membrane, and look at a lipid raft stain to identify whether RET is going to lipid rafts or not. We would predict that the mutants would not be going to these lipid rafts. And then finally, to kind of foray into some functional assays, I'd like to try a transwell assay. This could look at any invasion of these cells without ligand, but also with, and see if they are attracted more so towards the gradient ligand. So in this case, the cells would be seated up here and could either HEKs don't secrete any ligand, so they wouldn't have a gradient of attraction, or smooth muscle cells do secrete GDNF, so that would be a gradient for their attraction. And that would be an interesting comparison to try. So for some overall conclusions, I'm going to shamelessly plug my paper here. So for the TMEM project, we demonstrated that a potential role for TMEM in membrane stabilization. We identified that RET and other receptors could be potential targets in these TMEM mutant tumors. And then even more so, we could potentially expand the FDA approval of RET inhibitors to include these TMEM mutant tumors. So these inhibitors are approved for thyroid carcinomas. So if we could potentially expand that to include these tumors, which you wouldn't have previously thought would be useful. The MEN2 project we showed some constitutive localization to classroom structures, as well as some constitutive internalization and recycling. Preliminary data shows that high levels of endosomal compartment co-localization, as well as some associated signaling from those compartments. And our next steps, we aim to further explore MEN2 at the molecular level, as well as with the co-receptors to potentially identify areas for intervention. Here's some acknowledgements. So I'd like to thank my supervisor and supervisory committee, our collaborators in Texas or Toronto and Texas, as well as our current and past lab members.
Amazing. Questions for Tim? So you've got this nice knockout null cell line for the 127. In humans that have mutations, are they typically nulls or are they more subtle mutations? And so point mutations, that sort of thing.
Yeah, generally they have a complete loss of the wild type receptor.
So are there any interesting ones that are missense mutations that are more subtle in their phenotypes?
There are, yeah, there's different degrees. Some of them, they don't have quite as much. I know there are other forms that don't present quite as aggressively or quite as noticeably.
Where I'm going with this is, what is it doing? Do you know anything about how it's interacting with RET? How it's regulating its trafficking behavior?
Yeah, so I think it's not specific to RET. I think just by losing it, the membrane is kind of less regulated.
Yeah, you nicely showed EGFR and urines and a bunch of things. So it presumably is something that they all in common have when it comes to internalization and trafficking. So the mutants, though, 2A, 2B mutants, tend not to go eventually to the lysosome to the same extent as wild type. Do you have any idea why that is?
Yeah,
to your burning questions.
Yeah, because I think they're just kind of caught in a loop of trafficking. So they're constitutively active, and I think they're just constantly recycled back to the membrane.
So they're more likely to go into the recycling compartment than the lysosome.
That's my theory. Yeah.
Thank you for the wonderful presentation. I was just wondering if there are differences between CK and CB in terms of mutation burdens.
Mutation, sorry, abundance?
Mutation burdens. Some have low mutation burdens, while some have
Burden.
Yeah, so between 2A and 2B, the difference is based on the mutation.
That's a good question. I think I'll have to get back to you on that. Chris?
Yeah, thanks, Tim. So nice talking. There's a lot of data, so forgive me if I missed it, but with the TMEM mutant, what I thought was interesting, in addition to the other receptors you examined being sort of stuck at the membrane, if you will, is that you also mentioned there were changes in the membrane stability. So I wondered if those tumors are also more susceptible to other types of therapies because of the membrane instability that might also be able to treat them, but maybe it just hasn't been examined yet because the data has just come out. So do you know if other types of treatments have been explored for those types of tumors, and or do they have a lot of resistance to, like, are those tumors prone or less prone to resistance to treatment?
Yeah, that's an interesting thought. I think as far as I know, the Tima mutant theos are very rare. And there weren't really any treatments before that. They were kind of just resect, hope that it's gone. But that's an interesting, yeah, like maybe the penetration of the membrane would be a little bit easier. That's
Well, just you were talking about the lipid rafts as well being impacted, so that's why I was wondering about that.
yeah, that's interesting.
Thanks.
I have a sort of fundamental question I feel I should know the answer to, but hopefully you will. So what's the biological reason for endosomal recycling? I mean, I know that endosomes are also taking in things from their surrounding and recycling molecules like albumin, immunoglobulin. What's the point of recycling receptors? Does something special happen to those receptors?
Well, ultimately, when they're brought into the cell, the pHs are different within the endosomes, so they're dephosphorylated, and essentially, if the cell wants to prolong signaling, it will just recycle it back to the membrane for reactivation.
So they are remodeled, become okay. So they're dephosphorylated within the acidic endosome.
Yeah, and then just traffic back up if they want more signal, but if they have enough signal, they go and degrade them.
The co-receptor doesn't get recycled, did you say? GFR alpha?
Question, I believe it does. I'm not sure if we know that.
Okay, that'd be kind of
Probably, yeah.
interesting. Because...
Mhm.
And then you said sometimes, even in the endosome, it can signal. I think the mutant receptors can signal.
Yeah.
So how does that happen?
Yeah, so some previous work in our lab looked at ERK signaling specifically and found that it wasn't fully activated until it was kind of in the endosomes, and that it signals kind of off that membrane intracellularly.
Does it activate the same pathways?
I believe so. I think it's kind of for its full activity. That's what it requires, that internalization.
So, it's an interesting point, this idea of being able to use RET inhibitors on tumor, on, even 127 positive, but not necessarily RET inhibitors. Can that be extrapolated to, other receptor, lung cancer, I guess? So in lung cancer or other types of tumors where they're activated, RTKs, or they may be activated, but not necessarily always. Are there TMAM127 mutants that are associated with those tumors?
Yeah, so we know that TMA mutations also occur in renal cell carcinomas, and I believe that the receptor in that would be MET. So we haven't tested it, but we would figure that a MET inhibitor would help in renal cell carcinomas.
So another question about, so there are at least 3 RET inhibitors that you're using here, and I'm assuming all 3 are small molecules that are active site directed.
The sulfatinib and pralsetinib, they activate a different portion, because they were trying to get around
find a different location.
a different location. Yeah, so there's some gatekeeper mutations that came out of bandetinib, which is the multikinase. So then they developed sulfatinib and pralsetinib to bind.
Okay,
That's
that
another
answers my
site,
question.
yeah,
So
so they
gatekeeper
avoid that.
mutations, some of these molecules can circumvent gatekeeper.
Yes, but there's a couple other mutations that have been developed out of sulfatinib and pralsetinib that affect their binding.
Is there anything you can do with the cell to bias lysosomal degradation of the protein? I mean, you reach a point where there's a stochastic separation between recycling and degradation. Can you bias that pathway?
I believe we have a recycling inhibitor. The PhD person who I took over from, he tested kind of inhibiting lysosomal and proteasomal degradation. He kind of tested that. Sure, if we have a recycling inhibitor. I'll have to look into that, but yeah, that's an interesting thought.
Can you go back to your turf movie? That's noticed by that.
That's right.
So in this method, you're seeing the formation of an endosomal structure. But you were saying that, you can tell the difference between ones that are failed, that fall apart versus one that presumably get pinched off and endocytosed, because you lose visibility of the ones that get pinched off as well, right?
Yes.
So how do you tell the difference? between
Yeah, so I'm not the one that does that, so we have a script that actually is specific to clathrin coated pits. And it measures like if they don't reach a certain intensity, they don't qualify as an
As an
initiated
endosomal,
version. Yeah, so it's a MATLAB script.
Okay, so I can gather from that, so you would expect it reaches, a very bright spot and then it disappears, so there's some kinetics to the change in fluorescence that is indicative of that.
Yeah, I didn't do it by hand.
You didn't do it by hand. No. Other questions? Last chance. You won't give another talk for another 2 years. Okay, then join me in thanking Tim for wonderful.